It's my pleasure. I used to come there every year, almost for a few years, as guest of Walter Willett, Frank Hugh, Michelle Williams. But now it's nice to do this online. So let me bring up my slides. Can you all see it? Just a minute. Can you see my slides?
Yes, I do.
Okay, good. Okay, so this presentation in some parts will be somewhat controversial, but I want to make it that way. So, I mean, who are South Asians? These are people living in or with roots in India, Pakistan, Bangladesh, Sri Lanka, Nepal, Bhutan, Maldives. It's nearly 2 billion people, or a fourth of humanity. And it's also one of the fastest growing subpopulations in the United States. It's about 6 million people now and growing. So in terms of the pandemic of, when you look at the pandemic of diabetes, it's, I'm using the word pandemic here, every country in the world is affected. And the South Asian region is particularly India, and the South Asian region is pretty much at the epicenter, with already 82 million people in South Asia, expected to grow to 151 million. Then the issue was when in 1972, the prevalence of diabetes in urban India was 2.3%. In rural India was 1.5%. And the Indian diaspora or immigrants in many countries were studied in the next 20, 30 years. And as you see there, every place where they went to, the prevalence of Indians who migrated like me was much, much higher compared to the Indians who stayed in India. So this idea got entrenched in the literature that when Indians migrate to other countries, they acquire higher levels of diabetes prevalence or diabetes risk. And until this was challenged in a PhD thesis by one of my PhD students, Anjali Gujral, who basically asked the question, now with rapid economic growth in India, does that idea of immigrant Indians in other rich countries having higher prevalence than Indians in India still true? So she compared two cohorts, the CARS cohort, which is an NIH-funded cohort that we own in India. It's in Delhi and Chennai. This is data from Chennai. And the Masala cohort, which is a cohort run by my colleagues in California and Chicago. These are Indians born in America. So these are largely immigrant Indians. And the Masala was aged 40 and older. The CARS was 20 and older. So we restricted both samples to 20 and older. So as you see there, the first major finding was that in this study, in urban India, the prevalence of diabetes in 40 and older was much higher than the prevalence of diabetes in Indian Americans in Chicago and California. True for men, true for women. And then the general US prevalence. So you see two things here. Number one, overall, Indians are at higher risk compared to the US general average. And secondly, Indians in urban India have much higher prevalence than Indians in Chicago and California. You can argue about the reasons, but this flips the idea that it's only migrant Indians that have higher risk. And indeed, when you assemble all the prevalence studies in India in the period 1971 to 2015, you see the prevalence in urban India going up from 2.3 all the way to over 24%. And in rural India too, it's gone up from 1.5 to as high as 14, 15%. So clear secular increase in the prevalence of diabetes across India, if you like. So the question is, why do Indians have diabetes risk? And that's true even at low body indices, they have a high diabetes risk, which has led groups to come up with different cut points to define obesity in Indians and in Asians. Whether that's right or wrong, we can argue, but I want to raise some questions around it. This was another PhD thesis done by Rina Oza Frank, who looked at all the immigrant population in the United States. These data represent 36 million people. Immigrants to the United States does not include the undocumented workers, but it includes all the legal immigrants. And what you see here, when you plot the prevalence of diabetes by region of birth or region of origin, people from the Indian subcontinent and migrants from the African continent, these are not native-born African Americans, these are migrant African Americans. They have the highest diabetes risk overall across all immigrant groups. And when you draw this line which is this line is the prevalence of diabetes in obese European immigrants, and look at the Indian immigrants and the African immigrants. Even normal weight Indian immigrants, normal weight African immigrants have as high or even a higher diabetes prevalence compared to immigrant obese European immigrants or obese European immigrants. So why do South Asians have a high risk of diabetes? I put this talk together for an award lecture a couple of years ago. So when you review the literature over the past 40, 50 years, the dominant view in the literature is the following: that South Asians are genetically predisposed, they have more insulin resistance, they have higher visceral adiposity. And I am one of these people that likes to question any convention. So I said, let me ask, are these really true? Let's examine the literature and the current data to examine each of these questions. Firstly, I want to just summarize for you in one slide pretty much everything that we know that is useful about type 2 diabetes genetics. Again, it's a vast field, but I'm just summarizing it for you in one slide. There are about over 400 known SNPs that explain about 18% of all cases worldwide. Diabetes is a polygenic disease, except for some monogenic variants. But most of the SNPs for the polygenic variant of diabetes are not related to obesity and insulin resistance. That's something very important for you to remember. Second, the common variants of genes are very similar across populations, including Indians. And this was a classic paper published by Francis Collins and his team some years ago. Adding genomics data to simple measures do not improve prediction. This is from the Framingham Offspring study. And lifestyle interventions from all the large clinical trials, they almost negate the enhanced genetic risk of diabetes. I mean, this is an overgeneralization. There is clearly value for genes, genetic studies to understand etiology, to understand targets for intervention, precision diabetes, etc. But this is where the field stands. On the other hand, when you look at epigenetics, this study was done in Britain. They compared whites in Britain with comparable Indian Brits in Britain. What they found, there were 5 genetic loci that were associated with diabetes in both populations, but the epigenetic or the methylation score was much higher in the South Asians compared to the whites. So although there are no fixed gene differences, there might be epigenetic differences to explain the higher risk in South Asians. Now I want to step back and take you back to a few fun slides about the historic roots of diabetes in the Indian subcontinent. These are data from the anthropology group in Cambridge, who have studied skeletons across the world. This is the Mesolithic period, which is about 10,000 years ago. These are the heights of Western Europeans, male of about 160, females of about 150 centimeters. And this is 3 populations in Eastern Europe, males of about 170, females of about 160. And look at these 2 populations in South Asia. This is the Indus Valley population and the Gangetic Plain, men of 180 centimeters. That's about as high as the Dutch are now. And women are about 160, 170 centimeters. So 10,000 years ago, clearly looks like South Asians on average had higher heights. And what height is useful for, it's a good measure of maternal child nutrition and also has genetic influences. Barry Popkin and other colleagues constructed these data on changes in height over long periods of time. And this dark line is South Asia, the Indian subcontinent, where you see a gradual decline in height continuously over a long period of time and a little rapid during the period of colonization. But very lately, in the last 20 years, there's been some increase in the height. So something is going on there. So the other thing here is this, again, study from Cambridge. The average bone length in Indians has also declined over time, over the last 11,000 years. The Indians have less bone breadth, which is a measure of lean mass, compared to worldwide samples. So this the people at the bottom are Indians in Mesolithic period, historic, prehistoric, and modern across time. They generally have less bone breadth. And this is a more recent study comparing Indians' lean muscle mass of South Asians with Europeans. And what you see here, even in children aged 10 to 18, the lean mass in South Asians is much lower. almost 1.5 units lower, and it expands, it increases with age. This was a study in France comparing the autopsy organ sizes of Indians with French. Of course, these are selected specimens, but in general, Indians have lower BMI, lower weight, lower height, but much smaller pancreases, much smaller liver, much smaller lungs, much smaller heart. So all of these are interesting data. So the evidence over the past 10,000 years provides some context that the population of the Indian subcontinent has on average experienced a gradual decline of stature and lean mass, has on average poor organ development, including liver, pancreas, lungs. And the cause is speculative. It could be adaptation to climatic changes, other environmental factors, less food production with greater population increase. There are lots of theories about it, but this seems to be quite factual at the moment. And the other thing to remember is type 2 diabetes is really an ancient global disease, but it's meeting the 21st century lifestyle. You can go to the history of in Egypt, in Arabia, in Rome, in Indian texts. You can see descriptions of diabetes for over 2,000 years. And here is a lovely quote from a 6th century Indian textbook called the Charaka Samhita. There are 2 forms of diabetes, they called it. They called it Madhumeha or honey urine. One associated with emaciation, dehydration, polyuria, and lassitude, and the other with stout built, gluttony, obesity, and sleepiness. Remarkably, 2 different types, if you like, just through observations. But what is interesting now, when coming to today's talk, since 1971, you've seen a massive increase in the prevalence of diabetes. This is one city, but it's quite representative of major cities in India. And this period also coincides with rapid economic development, particularly from the 1990s across India. So now let's examine this question. Are Indians really insulin resistant, or could it be insulin deficiency in the population? Firstly, again, Anjali Gujral compared data from our CARS cohort in Chennai with whites in the enhanced sample for men and women by the 4 weight categories. And what you see here, when you compare Asian Indians with whites, at the higher BMI, yes, Asian Indians have a higher diabetes prevalence. But look at the Indians in underweight category and normal weight category, considerably higher prevalence compared to whites. Similar pattern in women. So this tells me while in the overweight and obese groups, the factors might be similar, the pathophysiology might be similar, and measurement could explain some of the differences, or fat distribution could explain some of the differences. In the very thin and underweight, are there other factors that we should be looking for? And indeed, this is like we could update this. When you map all the genes for type 2 diabetes and the genes for obesity, there is actually very little overlap. It means some overlap, but not a lot. Most of the genes for type 2 diabetes tend to be insulin secretion genes, but the genes for obesity tend to be insulin resistance genes. Now, I spent 4 years with the Pima Indians working for the NIH cohort from 1992 to 96. And that study has revealed a lot. The Pima Indians are a pre-Columbian migration to the Americas. They are thought to have arrived 35,000 years ago, somewhere from Central Asia. And steady, stable population, at least in the Gila River region, until in the early 60s, they noticed a sudden explosion of diabetes, which today we would classify as type 2. And it was about, by age 45, 50% of the Pima Indians were diabetic, and obesity was thought to be the driving factor, and rapid change in the environment was thought to be responsible. So this led to our hypothesis, if you like, that the primary driver was obesity, and insulin resistance was a dominant feature, and later, beta cell dysfunction began. So that was the kind of dominant hypothesis. So we said, now let's try and do a direct cross-sectional comparison of the Pima cohort with our CARS cohort in Chennai. These are cross-sectional, but I'm going to show you some longitudinal analysis as well towards the end of my talk. So what we found was quite interesting. the prevalence of diabetes in both populations, the Pimas and the Asian Indians, was very similar, 50% by age 55. But the obesity profile was very different. The Pimas were about 7 BMI units heavier and about 25 waist centimeters broader, bigger. The glucose profile, Pimas had higher 2-hour glucose, whereas the Asian Indians had higher fasting glucose. The Pimas were 2 to 4.5 times more insulin resistant than the Asian Indians across BMI and glucose strata. Even in the normal BMI, normal glucose strata, the Pimas were more insulin resistant, measured by HOMA-IR, whereas the Indians were secreting half to a third of insulin than Pimas across BMI and glucose strata. So we have done these studies in adults in India, children in India with diabetes, Indians in California and Chicago, and we find this consistent cross-sectional pattern of poor beta cell function, maybe occurring early, and maybe a more dominant impact on the progression of diabetes pathophysiology in Asian Indians. Just showing you one slide from Lisa Steinmetz, who got a PhD in this work. She looked at 1,200 people across glucose strata. And this is the beta cell function or disposition index declining very early in the natural history. And of course, this is insulin resistance going up with time. So all of these is offering us the hypothesis, should we be looking more closely at beta cell function in Asian Indians? And indeed, this is a cohort study in the United States comparing masala with the MESA cohort. These are non-diabetic people. This line here is South Asians. At all age groups, South Asians have poorer beta cell function compared to the other populations, and this dotted line is Chinese Americans. So Asians in general seem to have lower beta cell function compared to Blacks, whites, and Hispanics in the United States at all age groups, even in the non-diabetic populations. These are data from the New Delhi birth cohort, where they followed up children from birth till adulthood. So this green line are children who were born well-nourished. And then you start seeing their glucose trajectory. They seem to maintain good glucose trajectory until age 25. Whereas the children that were born low birth weight or who were stunted, their glucoses get bad by age 25. They develop IGT, IFG diabetes at a greater rate. And this paper had been published in the New England Journal of Medicine. And the authors at that time offered this hypothesis that those who went on to develop diabetes became obese relative to themselves. In other words, they had greater visceral adiposity, they had greater deposition of fat compared to muscle. We have a complementary hypothesis, not alternative, but a complementary hypothesis that those who went on to develop diabetes probably also have poorer beta cell function to start with. So this was a meta-analysis looking at the relationship between birth weight and type 2 diabetes and glycemic traits. It was a Mendelian randomization of 14, bringing together 49 studies, some 41,000 cases, 80,000 controls. For every standard deviation decrease in birth weight due to a genetic risk score, there was a 2.1 odds of type 2 diabetes. And this association was only present for fasting glucose, not for 2-hour glucose or for A1C. And this to me suggests something, because fasting glucose is a measure of poor secretion and hepatic insulin resistance. So one more clue that maybe poor secretion and hepatic insulin resistance, rather than any visceral adiposity, might be more related. And when they did a causal association using Mendelian randomization, it was stronger for the low BMI groups, again suggesting that maybe there is a phenotype of diabetes related to low undernutrition in childhood or maternal undernutrition that manifests itself differently. And with colleagues, we have done some studies looking more carefully, using more advanced, in smaller studies, using very good phenotyping. And what you find here, at men with low birth weight versus counterparts at age 19 to 20, they are shorter, they're lighter, they've reduced muscle mass, they have a trend towards lower mineral, bone mineral content, higher diastolic blood pressure, reduced dietary intake of protein, which again is a confounder, which we adjusted for. But the important thing, they did not show any difference in insulin sensitivity at age 20 years. Interesting. So again, this is the same group. This is personal communication. So these are unpublished data yet. So when you compare insulin secretion following a mixed meal challenge test, these are your, when you compare the people with diabetes, look at these two groups. The type 2 diabetes is type 1, and then the lean type 2, and then your overweight normal. In the lean and type 1 diabetes, insulin secretion seems to be much lower in general. So, so the other important observation that I've made is differences in response to treatment. When you bring together the vast body of lifestyle intervention studies, in general, South Asians, on the one hand, their progression to diabetes from IGT or IFD is much faster. It's about 12 to 18%. Whereas whereas in South Asians, it is much in the whites, it's much slower. In the whites, the reduction in terms of benefits from lifestyle intervention is about 50% reduction, whereas in South Asians, it's about 30%. Again, suggesting that lifestyle intervention works in South Asians, but might have a weaker response, largely because it works through insulin resistance, whereas the primary problem might be insulin secretion. And look at this trial, this data from the DREAM trial, which Salim Youssef did from Canada. It's an international study. The effect of rosiglitazone, the weakest effect was found in people from India, although it had benefits in terms of progression from IGT, IFG to diabetes. It was weakest in people in the Indians. However, when we look at the SGLT2 and DPP4, which largely work through beta cell function, etc., you find that Asians in general have greater response to those drugs like the DPP4 than the whites. This is a meta-analysis. And when you look at efficacy across Asian populations, look at South Asians. They have a greater decline in A1C for the equal dose of DPP4 compared to Chinese, and Koreans are also similar to Indians. So again, suggesting some ethnic differences in treatment response. Similar also to SGLT2, a bigger response in South Asians in terms of decrease in A1C compared to whites. So the question really is it insulin resistance or is it insulin deficiency? And let me share with you some recent data. This is an unpublished meta-analysis. It's in the final stages of getting published. This is from UK, where my colleagues compared South Asians to white Europeans. In general, South Asians had 2.5 times the risk of diabetes. They were on average 1 kilogram per meter squared lower in BMI. They had higher subcutaneous fat in men and no difference in women. But the interesting thing is, there was no difference in visceral fat in men and women, but there was higher liver fat in men by about 0.57 standard deviation, and also in women by about 0.3. So the bigger problem seems to be specifically liver fat and higher subcutaneous fat, not just general visceral adiposity. And subcutaneous fat is quite harmless, whereas liver fat is extremely harmful for cardiometabolic health. So this is a paper published in the Annals of Internal Medicine. Again, looking at different weight groups and looking cardiometabolic abnormalities like high glucose, high triglyceride, low HDL, etc. This is a group of people with normal weight. Look at the South Asians. 52% of South Asians in the normal weight have some metabolic abnormality. It's also present in whites, but that's about 18%, suggesting that this low weight cardiometabolic abnormality phenotype is present in all ethnic groups, but it's more frequent in South Asians than in others. And these data, by the way, were adjusted for VAT. So it's not, so it takes the metabolic, the things over and above, fat distribution suggests that there might be other factors. What can it be? When we quantify hepatocellular lipids by diabetes, look at type 2 diabetes. Look at the huge amount of hepatocellular lipids, compared to type 1. And this is malnutrition-related diabetes, small subsamples in India. And you find the same thing in terms of pancreatic fat deposition in type 2 and in malnutrition-related diabetes in India. These are detailed studies using MRI, etc. So this was a recent paper just published earlier this year, a longitudinal comparison of two cohorts, the Pima Indian, the South Asians versus blacks and whites from the ARIC cohort. Firstly, look at the group under BMI of under 25. South Asians have a higher incidence compared to US whites and US blacks. It's true for men, true for women, and overall, so that's the first important finding. More importantly, when you compare HOMA-IR and HOMA-B, that's the South Asians, that's blacks, and that's US whites. And then this is HOMA-B, that's South Asians, that's blacks, that's whites. So this is after adjusting for age and BMI, suggesting that South Asians are actually on average less insulin resistant than US blacks, and slightly lower than white, but although not significantly. But in terms of beta cell function, South Asians are just generally seem to be lower than blacks, comparably, and about as much as whites in the United States. So in our models, when you compare standardized regression, South Asian, the HOMA-IR seems to be a more stronger predictor of diabetes progression in blacks and whites than in South Asians, whereas HOMA-B seems to be a stronger predictor in South Asians compared to blacks and whites. So we also did a comparison of young Pimas, 20 to 44, with young South Asians. And the reason we restricted to age 20 to 44 is because after the age of 45, there's hardly any diabetes, new diabetes, incident diabetes in Pimas. And what you see here, in the lower BMI groups, less than 25, less than 30, the South Asians have much higher incidence compared to Pima Indians. And okay, both for men and for women. And then when you compare insulin resistance, HOMA-IR, the South Asians are considerably lower insulin resistant than Pima Indians, whereas the South Asians are also secreting less insulin than Pimas, which raises an interesting question why that is the case. But when we run the models again, once again, insulin resistance seems to be a slightly stronger predictor in Pimas, whereas the insulin secretion seems to be quite similar in the two populations. So the question now really is it now time to consider heterogeneity of type 2 diabetes? And this will be my last 10 minutes or 12 minutes of my talk. So some years ago, 5 years ago, I received the Kelly West Award for Scientific Excellence at the American Diabetes Association. So at that time, using the cross-sectional data and our small studies, I offered a theoretical construct. I said, perhaps there are at least two types of type 2 diabetes. One, related to high metabolic load, I called it 2A. The primary problem is poor insulin action. The causes are adiposity, physical inactivity, dietary factors. The diagnosis is probably a 2R glucose. The initial treatment is probably your lifestyle, metformin, etc. And the classic ethnic type might be your Pima Indians. But then I hazard the guess that perhaps there's a second type, which is type 2B, which is poor due to poor metabolic capacity. But the primary problem is poor secretion to start with. Notice both these bars overlap. So both the variants that I thought of at that time had both the problems. The question was which problem was dominant, which started first. Was it insulin secretion or impaired insulin action? The causes is where it gets troublesome. We really don't know the causes of this type. It could be poor maternal child nutrition. It could be reduced beta cell mass. It could be microbiomes, lipotoxicity, endocrine disruption, or all of the above. And the diagnosis might be a fasting glucose or a 30-minute glucose, post-challenge glucose, because it measures first phase insulin response. The treatment might be a little bit of lifestyle might work, but secretagogues, gliptins, insulin might be a better treatment here. And your classical phenotype might be Asian Indians and all of the populations somewhere in between. So all populations may have both the phenotypes. The question is, which is more frequent? And indeed, when we did another study comparing a 3-step glucose tolerance test, we found 4 clusters of diabetes. And we were particularly intrigued by this cluster in purple, which has a normal fasting glucose, a normal 2R glucose, but a high 30-minute glucose. And this group was about 22% of the population. and was at 9 times the risk of developing, or 12 times the risk of developing diabetes compared to the group that was normal. So again, giving us greater data to argue for phenotypes. But the classic study here arose from Scandinavia, where they had a fantastic biobank. And when they ran their data, they came up with 5 distinct phenotypes of diabetes. The first 2 types were related to poor insulin secretion, and the next 3 was related to poor insulin action or insulin resistance. This group type was largely entirely type 1, but this group here, which was insulin deficient but relatively low BMI, seems quite similar to the variant that we are finding quite frequently in South Asia. Whereas these 3 groups, one was related to aging, one was largely related to obesity, one was somewhere in between. And these studies have been now replicated in several populations, including in Chennai in a large data set by Dr. Mohan. And they came up with 4 of the 5 phenotypes the Scandinavians found. They didn't find much of type 1 in India. There wasn't. They excluded that from the analysis. But they found this group. In the Scandinavian, this was about 7% of the people with diabetes, whereas in South Asia, this was like 26%. And then the other 3 groups were quite similar, but less frequent. The age-related was quite similar too. So this phenotype, this insulin deficient variety, was something they found in more than a quarter. Alka Kanaya repeated the study in across ethnic groups in the United States, in whites, in blacks, in Latinos, Chinese, and South Asians. And I want you to look at this one in orange, which is your insulin deficient thin people variant, what they call severe hyperglycemia. In South Asians, it's about 38%. In your whites, it's about 11%. This tells us that these phenotypes are probably present in all ethnic groups, but the frequency of them is what varies. And in South Asia, there might be 30 to 40% who are thin but have insulin deficiency, and that might be related to historical malnutrition or maternal child malnutrition, or to beta cell dysfunction due to other causes. And just to complete my talk, my colleague in Vanderbilt, through using organ donations, in the United States, they've been studying beta cell function in autopsy specimens of people with type 1 diabetes who have died with type 1 or type 2 diabetes. And what they find is very intriguing. In type 1 diabetic beta cells, that beta cell function is normal, interestingly, whereas what may be happening is it's autoimmune damage of the beta cell during life that impairs secretion. Whereas look at this, in type 2 diabetic, pancreases, autopsy cells, these are normal, type 2 on oral agents, type 2 on insulin, and a graded response suggesting greater decline in insulin secretion. So once again, this classic idea of type 1 being poor secretion, type 2 being insulin resistant, may get flipped around. It might be, we might need to pay greater attention to type 1 as a poor insulin secretion problem. And this was a recent study from Japan, where they looked at, actually measured beta cell mass using mass spectrometry and MRI, etc. So in non-diabetic individuals, they compared them with diabetic individuals. In diabetic individuals, there was less beta cell mass, less beta cell number, greater apoptosis, greater dedifferentiation and degranulation. We are trying to set up the study in Hyderabad to look at autopsy specimens of Asian Indians with type 2 diabetes. So perhaps we need to be thinking of type 2 diabetes along the spectrum of knowing the islet cells, the alpha and beta cells better, doing multi-omic profiling, especially single cell profiling, data integration, to better understand the pathophysiology. So coming back to my questions, why do South Asians have a high risk of diabetes and low BMI? The dominant view in the literature versus the evidence that I've shared with you, the dominant view in the literature is South Asians are genetically predisposed, but I think so far there's no difference in fixed genes, at least in major genes. There might be in minor variants, I don't know, but there might be a bigger problem with epigenetics. the dominant view in the literature is South Asians are more insulin resistant, but I think no, they may be on average be more insulin deficient to start with for whatever reason. The dominant view in the literature is they have high visceral adiposity. I think no, they have higher hepatic adiposity and greater subcutaneous adiposity, which may have different pathophysiological effects. So, why do South Asians? I mean, evidence suggests that they are more insulin deficient than insulin resistant. The insulin deficient phenotypes may be more prevalent in South Asians. They display a propensity to high ectopic fat deposition and pancreatic fat deposition than overall adiposity. They respond less to interventions that alter insulin sensitivity and insulin resistance. They respond better to interventions that target beta cell dysfunction. So putting all this together, we have a hypothesis paper published in Diabetologia. Alka Kanaya and I hypothesized that maybe there are two problems in South Asians. Number one, problems that are evolutionarily determined over thousands of years, hundreds and thousands of years, which has given them innate poor beta cell function, low lean muscle mass, which impairs insulin secretion. And then in response to contemporary or modern lifestyle, they have greater ectopic fat deposition in the liver and muscle, which impairs insulin action. So when you combine these two, there is a rapid progression of type 2 diabetes in South Asians. So I want to thank, our center does work both domestically in the United States, we're an NIDDK funded center, and in South Asia, we are hoping to have a very highly funded program Project Grant, which is going to fund a large precision CARS cohort in India. I want to thank all my colleagues, thank all my funders from several sources, and I'll stop there and take any questions.